Ed Pryzdial. Anybody know him? No.
Yes.
Here we do. Okay, so Ed, well known to the CBR and to UBC. He did his PhD in biochemistry and complement in Toronto, and then went to Vermont and did more biochemistry and coagulation and fibrinolysis, which he has stuck with. Went back to Ottawa, right? To the Canadian Red Cross, where he was a scientist, and then they moved him west to Vancouver, where he's been a leading figure as a scientist, so with the Canadian Blood Services, an integral member of the Centre for Blood Research, member of the Pathology Laboratory Department, and a good friend, colleague. He's an amazing writer and incredible artist, and you'll see it today in his science. Ed, all yours.
Thank you very much for those kind words. I'm just setting my timer because too many of you know I can go on and on and on. Okay, timer done. I've been asked to talk about dengue virus and thrombocytopenia, and I'm really pleased to do so, but in particular, I'm honored also to be doing so because I'm going to be talking about the work of a really great grad student, Sideh Tegen, who is PhD 2B status. Why do we care about dengue virus anyway? Well, dengue is kind of bad. Its infection rate is huge globally. The numbers that you're seeing here now, I really should have updated for you, but they're on the rise. And they're on the rise for lots and lots of reasons, which we're not going to touch upon today. But about a 500 million infections occur every year. About 100 million, more than 100 million of these result in some serious sicknesses. And when we're talking about sicknesses in dengue virus, the average sickness is like, everybody here has had the flu. Picture flu 10 times worse. It results in bone aches and really serious kind of sickness like flu-type sickness. And then when you get to severe illness in dengue virus, well, check this out. Oh, darn. You bleed from the eyes. Come on, I want to go back. How do you go back on this thing? Come on, wait, save me. You bleed from the eyeballs. What's going on? You bleed from the eyeballs.
Just go like this. Don't look at that. Okay?
Yeah. Freaking Mac. Did I say that loud? Okay. And how do I use the mouse?
I didn't know I had to give you a lesson.
I want to use a mouse.
Yeah, just go like that.
Okay, so how come it's screwing me
When
over?
it disappears, and you go like this. Okay.
Okay.
Do you want me to give you a
No,
try?
but I don't want that.
Don't push hard, just touch.
Okay. You screwed me over here, Conway.
Time.
Okay. Oh, I've got some choice words for you later. And notice with this bleeding, we're very interested in coagulation in my laboratory. So this is another reason why we're personally interested in dengue virus, because of all of the links it has to hemostasis. With these severe illnesses, it can induce fatalities. About 50,000 occur per year. So we really need to have to understand what causes these links to bleeding in order to understand some treatments. And there truly is not any good treatments. So what we have, probably the best treatment that we have, is linked here. And because I've flipped around a little bit with the slides, you may have seen the answer already, but if you can raise your hand and know the answer of how this particular excellent treatment works, you get a piece of chocolate. So who knows how this treatment works? Well, you weren't paying attention to the damn slides as they were going by. This is the active ingredient in mosquito repellent. I'm going to give this piece of chocolate to Katerina because she is a hard act to follow. And she gets to have this on her flight home to Toronto. All right, deal? Okay. This is bug spray, and dengue virus is a mosquito-borne vector. Is a mosquito-transmitted disease, a mosquito-transmitted virus, and it really exists predominantly in the tropical and subtropical regions of the world. Canada, luckily, does not have the mosquito that carries this virus, but it has been identified in the southern tips of Ontario. It's not endemic. It has been identified, but it is spreading. It is spreading north. It exists in the southern regions of the United States. It didn't 20 years ago, but it's spreading. So we got to be on guard. What we care about with this virus is that part of the bleeding manifestation is caused by a decrease in platelets. Every single case, whether it's severe, not severe, asymptomatic, has a decrease in platelets. And there has been lots of reasons why, one of which is loss of platelet function as a consequence of the virus having antibodies decorating it, and then those antibody FC receptors attaching to the FC receptors on the platelet surface, and so the platelet gets cleared as a consequence in that mechanism. Also, we can have a decrease in the number of platelets that get generated just because the virus loves to infect megakaryocytes, which is the precursor cell in marrow that produces the platelet. We're going to talk a lot more about that because today is infecting megakaryocytes to investigate further certain mechanisms. There's also a mechanism where antibodies to a virus-encoded protein gets secreted by infected cells into plasma. The most abundant and possibly one of the only antigens that gets secreted is something called NS1, a virus-encoded antigen. And the antibodies that get formed to this virus-encoded antigen cross-react to a host-encoded protein called PDI, protein disulfide isomerase, for anybody who cares. And that exists on platelets and megakaryocytes and other cells as well. But if this antibody to NS1 can cross-react to a host-encoded protein on a platelet, it's going to get cleared. Well, we know a little bit of something of this protein. NS1 can exist inside of cells. Oh, it's doing it again. I'm too stupid with your damn computer. Okay. It can exist inside of cells in a monomeric and a dimeric form. And it can also exist outside of cells in a dimeric form. And it can also exist as a hexamer. In the dimeric form, it can have this glycosyl phosphodienostatide linked sticky foot, where it can bind again to the outside of cells. And as a hexamer, it can bind to heparan sulfate proteoglycan. Well, people have never considered that this virus antigen can stick to the outside of cells. So why do we have to have this indirect mechanism? Why can't the immune system see an antigen on the outside of cells? And so our hypothesis is a direct immune recognition of this antigen on the outside of cell, and that's what Sade is hunting for, this antigen on the outside of megakaryocytes, because we already know that this virus can infect megakaryocytes. From our earlier work, we also know that this virus can infect platelets. What we would expect to see if we did some immunofluorescence then is that the virus would have a nice glow over, the cell would have a nice glow over its surface, a corona, if you will, decorated by NS1 over its extent. What we have seen, as I said before, is that platelets can indeed express NS1. What Sadeh has recently shown is that so can megakaryocytes. The difference between these two experiments is that this is total NS1, what's been secreted as well as what's on the surface as well as what's inside of platelets, whereas this is only what's on the surface and what's inside of platelets, not what's secreted. So what Sadeh wanted to do is she wanted to take a look at what's only on the surface. And remember, over here, NS1 can be on the inside. So if we do any antibody type detection of what's on the surface, we want to make sure that the antibody can't get in. So we're going to try to look for cells that have not been broken up in any way for the antibody to get in. So we want to look only at intact cells. So the way that we're doing that, only looking at intact cells by flow cytometry here, is using something called a live dead assay. It's a commercial assay designed to make sure that the cell is not busted open. So up here you can see that these are megakaryocytes. You can't see that, but these are megakaryocytes that have clear distinction between permeabilized, where the antibody can get inside, and intact, where the antibody can't get inside. So we can make that distinction. These would be megakaryocytes, and these would be probably platelet-like particles coming off of the megakaryocytes. And you can see that after 10 days of culturing these megakaryocytes, and these aren't really megakaryocytes, these are megakaryocyte-like cells. It's a cell line called MEG01, which we're using as our model cells. After 10 days of infection, there's fewer intact cells because the virus really messes up the cells after 10 days. There's only 47% after 69% of 10 days of culture. Bottom line, only looking at the intact cells, we see an increase of surface NS1 on megakaryocytes. So indeed, virus antigen is on megakaryocytes, poisoning the megakaryocytes for recognition by the immune response and subsequent clearance, explaining, in part, thrombocytopenia, decreased platelet. We have also got some help by a really excellent postdoc research associate that was with us for a very brief time. He did a similar experiment for us on platelets, showing platelet surface also has NS1 on them. Eventually that died off, and we don't know why that is yet. So they then asked the question, well, we know it's on the surface, and we know these cells are starting to get a little bit ratty because of culture as well as because of virus infection. What do they look like? So she did some immunofluorescent experiments. Remember, we're expecting to see a lovely green corona of NS1 on the surface of these cells, the intact cell. This is what they looked like. So we've got an intact cell. Okay, we've got an intact cell here, and then we expect to see some cells that have been infected and probably pulverized a little bit by these viruses. Here is a fluorescent micrograph of what these cells looked like. In green is the NS1. So you can see that the cell distribution of the NS1 distribution on these various cells is not homogeneous. It's not that lovely little corona that I was thinking might exist. In some cases, there's great big blobs of NS1. In blue is DNA. And in this case, we're checking for permeability using a red marker showing where mitochondria are. So red means that the antibody was able to get into the cell to show us where a mitochondria might be. So let's take a look at this. So this means that there's probably multiple ways that NS1 is stacking up on the cell, and there might be multiple ways that even DNA and NS1 might be seeing each other. So here they are kind of broken down. There might be some cells that have neither NS1 or DNA on their surface, maybe just NS1 on the surface, maybe just DNA on the surface, and maybe both. There's some real strange looking configurations of NS1 that we did not predict. Let's just look at this one and enlarge it a bit so that you can see it a little bit more clearly. This is not a halo. That's some kind of an NS1 aggregate mixed in with DNA. You know, when I think of DNA coming out of a cell, I think of a net. This is not a net, clearly not a net, which is a long, spindly thing that looks like it has just shot out of a cell. That's not what this DNA configuration reminds me of, but it's reminiscent of DNA presenting NS1 somehow. I'm not going to venture out and say that that's what's happening because we don't know that, but it's food for thought. Bottom line, this is way more complicated than we thought it would be, and very interesting. Well, while we were having one of our weekly meetings, I said, well, you've got to quantify these various populations and configurations of NS1 and DNA on the cell. So that's what she did. She sat there and she quantified, and Sade was not happy with me. But she did it. She came back, her eyeballs were a little crossed. She was frowning. She slapped these data in front of me and she said, well, here they are. The majority of cells expressed NS1 on their surface solo. These are intact cells we're looking at. Very few had DNA on their surface. A few of them had DNA plus NS1 presented simultaneously. These are busted up cells. Well, now busted up is probably a stretch. These are cells that were permeable. Similarly, most of them had NS1 alone on the surface. Very few had DNA alone, and a few of them had the combination of DNA plus NS1. Bottom line, there's lots and lots of cells with NS1 on the surface poised to tickle the immune response. Immune response means complement. Right, Irene? That's what we looked for. Irene told you earlier that the big boss of complement is the deposition of C3b. So that's what we searched for. Eileen told us that if you have complement deposition on a cell or on a particle, it is either going to be cleared by phagocytic cells or it's going to be exploded by the membrane attack complex. So all we did is we looked for C3b deposition. Here we checked for classical pathway, where we took patient plasma, we treated megakaryocytes that had been infected for 10 days, because as you saw in that graph, we have lots of NS1 expression after 10 days post-infection. And then we induced by treating with serum, where there is a wonderful source of complement proteins. And then we followed C3b deposition using an antibody and flow cytometry. We got sources of dengue virus infected patient plasmas, or sera actually. And with the help of a wonderful postdoc in our laboratory, Mohamedou Rashid, he helped Sadeh design an ELISA to test these samples for whether they had specific antibodies to NS1. And these did. All of these samples did. And you can see, compared to our myriad of controls, oops, yeah, I'm doing it again. Our myriad of controls, there was a lot more complement deposition. These did not have specific NS1 antibodies, but obviously they have antibodies to other dengue antigens. They still had an enhancement of complement deposition, as would be expected since they're dengue positive. So what we have, quite clearly, is we have C3 deposition. These megakaryocytes are ready to be cleared by the innate immune system. Then what we did is we took one of these patient samples. It just so happened to be the one that we had the most sample of. We ran it a bunch of times, and then we pre-added actually to the patient sample purified NS1 to inhibit or to fill up the antibody antigen combining site to test the specificity that that's what it was recognizing on the megakaryocyte. And we inhibited complement deposition. So this tests the specificity that that's exactly what it was recognizing. So we've got NS1 specific deposition testing our model that NS1 on the surface of these cells is doing what we think it was doing. Remember I told you about that cross-reactivity business between anti-NS1 patient antibodies and protein disulfide isomerase, PDI? We wanted to make sure that the antibodies we're using, or the antibody in that patient sample, was not recognizing PDI. And so using that antibody that Sadeh and Mohammed developed, they put NS1 down on the well, and they came along with either the patient antibody or the antibody we used in the flow cytometry, pre-treated these with either purified NS1 or purified PDI to inhibit the interaction with the NS1 that was put down on the well. And what they found is that this antibody here to NS1 was beautifully inhibited by NS1, but not by PDI, which means it's not cross-reactive. Then they came along with the patient anti-NS1, showed that it was beautifully inhibited by purified NS1, as expected, but not by PDI, which means it's not cross-reactive too. So the idea that's in the literature of this cross-reactivity, at least in our model, does not apply. So we still have this idea that NS1 on the surface of megakaryocytes is a viable complement initiator. Alternative pathway of complement We just looked at the classical pathway, which is antibody dependent. The alternative pathway is not antibody dependent. We just took virus-infected megakaryocytes, has NS1 on the surface. Remember, these are 10-day-old cultured cells, which may also induce alternative pathway because they're becoming apoptotic. And then we followed C3b deposition. So here's uninfected cells compared to control, which induce the alternative pathway. Here's infected cells, which induce even more C3 deposition. So virus is inducing alternative pathway deposition of C3 on these cells as well. And over here, what we did is we inhibited that virus-induced deposition using an alternative pathway therapeutic inhibitor. This is a one-shot concentration. You can see that there is some inhibition going on, suggesting a mechanism to reduce thrombocytopenia using a complement therapeutic called danicopan. So what I've told you about, virus first binds to the receptor on a cell, goes into the cell, replicates, it leaves the cell. That's a virus life cycle. In the process, it makes a bunch of virus proteins. I think that this could be a general virus mechanism, which is why I said virus here. Of course, I've talked about dengue virus, which makes NS1. I showed you that there are several configurations or different modes of virus NS1 that might exist on the cell. Big blobs, aggregates of NS1 may exist. We don't know what kind of receptor-mediated mechanism is holding that on the cell surface. It can exist as possibly a dimer, GPI-linked, or NS1 hexamer, HSP-linked. This NS1 is triggering classical pathway mediated C3b deposition as well as alternative pathway mediated deposition. And this is suggesting that we have therapeutic options that are already undergoing some thinking in the literature and in the clinic using various complement therapeutics. But we have to think also about the use of vaccines. Vaccine development is against NS1 currently. I don't think it would be a good idea to develop an antibody against NS1 if we know now that NS1 exists on the surface of platelets and megakaryocytes. We have to think about this very carefully. She's the one. Michael has been involved in this since day one, and Mahmoud and Kalyan have also been involved in this project, helping out today. And also helping out me, our wonderful outdoors. My grandson, sitting here on Spanish Bank. This is the view out of my backyard. And here's our lab.
Thank you. Very great talk. So for the experiments that you showed, you had DNA on NS1. How do you know they were DNA and not generally nucleic acids, maybe RNA?
I have no clue. That is nucleic acid type material. That is, yeah, you're dead on. I only presumed because of the amount that was stained.
And is it possible that they could cross-link that nucleic acid could cluster in NS1?
Yes. There are multiple mechanisms that we would love to investigate. Today is wrapping up, and I'm hoping that I'll be able to follow up her studies with more detailed experiments concerning potential direct interactions between DNA and NS1. If I think that answers your question.
Yeah, thank you.
Great talk, Dr. Pryzdial.
Hi, Steve.
Hi. I have a question about, is there an advantage for the virus for the NS1 to be exposed on the platelet surface?
I honestly have not considered that question at all. I have no clue. We can guess. We can certainly guess that. I'm not even going to.
Over here, Ed. Nice talk. Thank you for that. I'm wondering if you've thought about or are thinking about using either recombinant NS1 or if you could purify out NS1 from a virus-infected like supernatant and then treat platelets so that you essentially separate the NS1 from the virus, just to see if you get a similar complement response, for instance.
I'm not sure I get it.
So, like some of these cells,
you can even buy NS1, recombinant NS1. I'm not sure I understand your question.
Yeah, just so, because like some of those cells looked like they were dying from virus infection, right? So, like, would you get a similar complement response if you had just the cells binding to NS1, platelets or megakaryocytes binding to NS1 in the absence of the virus, right? Would you still get a similar complement response, or?
There were in some of those controls. This is a beautiful little experiment because you can set it up so that cells are not even infected, and the answer is those cells can induce a complement response because they're undergoing apoptosis. That is good enough to induce a complement-mediated response. They do not undergo as strong a complement-mediated response in the absence of virus.
Okay, thanks.
So I have a question for you. Several years ago, must have been, I don't know, at least 10 years ago, there was a paper about the hemorrhagic or thrombocytopenic manifestations of dengue requiring a second infection. So are we seeing these patients having antibody-induced thrombocytopenia, basically ITP, but they have also complement activation?
I'm not sure of the status of those patients. I think that the current thinking is that about 40% of patients can be explained by antibody-dependent enhancement, the mechanism you're describing. And again, I'm not sure what the status is of those particular patients, the severity of their disease, and whether they escalated to severe disease. All of them for sure were NS1 positive and or virus positive. That's all I know about them right now.
The other thing is that the NS1 binds by the GPI anchor. Is that correct?
NS1, that, or in the hexameric form, it can bind to heparan sulfate proteoglycan.
So one of the thoughts would be, what if you took megakaryocytes from patients with PNH, where they've lost their GPI anchor?
We could try that. That would be a rare person.
Yeah, I
But
mean,
yes,
I'm not saying
we can-
that's the
we can
regular
do that.
But what would you see differently? Would you see loss of binding?
Don't know. We don't know, we do not know how that NS1 is binding back. One perhaps even easier experiment that we are in the process of attempting is to ask the other question. How much of that is binding to heparin sulfate proteoglycan? We sprinkle the experiment with a little heparin.
Right.
Yeah, you know, we have to do both.
Interesting.